So welcome in, everybody. Good morning, good evening, good afternoon, good night, depending on where you are. Before we get started and I introduce today's speaker, I just want to make a couple of administrative announcements. Next month, we will be having our Cancer Cachexia Society business meeting on October 13th. It's a Wednesday at 12 p.m. Eastern Daylight Time, which Cancer Cachexia Society members will get an email with a link to join us. It'll be by Zoom, just like this. Dr. Teresa Zimmers is going to tell us a little bit about her vision for the society as our new president, and we'll talk a little bit about things the Education Committee has planned for the next year, with, I believe, some also input from our Patient Advocate Committee. And then after that, in November, we're actually going to try something a little bit different. For our education seminar, we're actually going to have a debate. So Fabia Penna is going to host two outstanding scientists, and we're going to talk about fasting mimicking diets and nutritional support for cancer patients. And so that is actually going to be on Tuesday, November 9th, so our usual 12 p.m. EDT time, but a Tuesday instead of a Wednesday. And you'll also get an email and a Zoom link from that. So if you don't get the emails, feel free to put in the chat that you need them, and I can correct that. So it is my pleasure to introduce our speaker today. Dr. Aaron Grossberg is an MD, PhD scientist from Oregon Health Sciences University, and he is a professor in radiation medicine. He first got his bachelor's degree at the University of Colorado Boulder, and then earned both his MD and PhD degrees from Oregon Health Sciences. After additional training at Providence St. Vincent's Medical Center and then MD Anderson, he returned to start his independent career at OHSU. He has a clinical practice that focuses both on breast cancer and pancreas cancer, with particular interest in how radiation can be treated or can be used to treat those cancers. And he has research interests in both early detection and effective treatment of cancer by radiation. But what he's going to tell us about today is his interest in the whole body. And so as a physiologist at heart, it always warms my heart when researchers are really interested in the broad view of patients and how cancer affects these patients. He's been incredibly successful early in his career. He's funded by an NCI K08, which are not easy to come by. And so we're really thrilled, Dr. Grossberg, to have you here today. After Dr. Grossberg's talk, I will field questions. You're welcome to put them in the chat. And if we have some time, we may also have some people unmute and have a discussion. So without further ado, take it away, Dr. Grossberg, and thanks so much for being here with us.
Thanks for that great introduction. And I'd like to also thank Dr. Zimmers for inviting me to join you guys. Looking forward to starting, to getting involved with this group. As a junior investigator, we're all trying to do the same thing here. And I suspect that all of us have a little piece of this pie figured out. And if we can get our pieces together, maybe we can make the whole pie and make some real differences in patients' lives. And I think I try to keep my focus on that. As much fun as the game of science is to play, to remind myself that that's what we're actually headed for. So, I am also a physiologist at heart, which makes me a bit of an odd duck in radiation medicine, where we have lots of physicists and now budding immunologists. But for me, it really cancer has always been a systemic disease, or at least the interest has always been a systemic interest, and in particular, the ideas of cachexia and metabolic shifting that occurs as a result of cancer and how that impacts our patients. And so, today, I'm going to talk a little bit about some of the work. Much of this work is the work funded by the K08, and some of it is sort of the purview of some other grants. I don't believe anything I'm going to show today is published, so we have that to look forward to. I'm very much open to questions and being challenged on this stuff, because we want to get it right. So I have nothing to disclose. Hopefully that'll change when we figure out how to cure cachexia and have to make those arrangements. At OHSU, we've sort of aligned ourselves with a goal of a world freed from the burden of cancer. And what we talk about here is not just the disease itself. Most people talk about cure, but in fact, what we talk about in cachexia research, because this is such a major burden to our patients and their caregivers. And I probably spend the majority of my time, especially with my pancreatic cancer, my patients with pancreatic cancer, talking to them and their loved ones about nutrition, about weight loss, about weight gain, and their anxieties therein. And I think it really shows that even though that's something that we have very little data that we can help much with, it still remains the center point of many of our conversations. So cachexia, I don't have to introduce this to everyone here, but as you know, it's really characterized by this pervasive and persistent muscle and fat wasting, behaviorally with fatigue and anorexia. And we care about it because it impacts survival, it impacts quality of life, and really impacts tolerance of therapy. And in fact, I think that for the most part, cachectic patients are almost universally excluded from clinical trials. That's getting better now with more focus on those patients we don't include. But that's really hard when we're in the clinic and we want to assign a therapy. And the truth is the patient that sits in front of us was never included in the trial that tells us that therapy works. And sometimes, we cause more harm than good in that setting. So, that's why, we remain interested in this. And in pancreatic cancer, it's particularly interesting because about half of patients present with cachexia at the time of their diagnosis. And over the course of their treatment or their cancer journey, over 80% will develop cachexia. And in pancreatic cancer, as we'll talk about in a minute, it really is multifactorial. And almost all the treatments that we have for pancreatic cancer really exacerbate these metabolic issues. And so you're starting from a hard place and headed towards a more difficult place. And so the urgency in this disease seems to me to be extraordinarily high. Now, we know that one of the key features of cachexia is that it seems to be refractory to nutritional supplementation. And one of the thoughts as to why that is the idea that that these patients are in a hypercatabolic state. It's sometimes hard to define what that means, but I think a lot of this is based on both clinical and animal data that clearly shows that cytokines are independently capable of causing wasting, that they are capable of causing an increase in resting metabolism, and certainly some small studies in humans that show that some populations of cachectic patients are indeed in a state of elevated metabolic rate. And the idea being that perhaps the body is then titrating that metabolic rate to stay ahead of whatever nutritional input there is. But a lot of these data, despite this, anti-cytokine therapies have generally been ineffective in clinical trials. And some of that may be the sort of biblical nature of cytokines, how one begets the next. But some of it may also be that it's multifactorial. And some of it may be that indeed cachexia has some other etiologies that probably are worth exploring. But a lot of these data were derived really when you think about can you guys see this bar at the bottom of my screen? Is that blocking the picture at all, or is that just visible to me?
No, but I think just you have the bar.
Okay, all right, thanks. I was just afraid it was covering up some of the. Cachexia is not a binary state, is becoming more and more obvious, and we think about it as pre-cachexia, sort of cachexia writ large, and then a refractory state, at which point we probably can't recover patients. And a lot of the data that we have is generated by looking at patients in this sort of refractory, patients or animals in this sort of refractory state, and then comparing them to healthy controls. And the question that I've always had is, how do they get from here to here? Our definition of cachexia mandates that you can't really call it cachexia until at least a substantial amount of wasting has happened. And the question is before they're there, what is that driving biology? And is that biology the same as the biology that we're seeing here at the end? And so that concept has really motivated a lot of the work that I'm about to show, and trying to understand what these early events are in the development of cachexia and how we end up in a state where now we are depleted in terms of our adipose and fat mass. When I said pancreatic cancer cachexia was multifactorial, what I was really talking about is you can have any of or all of these going on in any given patient, and each independently could cause cachexia. So we know there's anorexia and nausea that can be caused by perineoplastic processes, inflammation, or even gastric outlet obstruction causing, distension of the stomach. We know that gastric outlet obstruction itself can prevent food from transiting through the GI tract and diminish nutritional availability. We know that exocrine insufficiency is a hallmark of some of these patients and that they're malabsorbing, and we see that with the advantages that Creon offers patients in terms of absorption, but this remains a major issue. And then finally, there is the idea of perineoplastic processes that induce lipolysis and proteolysis directly. And I think this has received most of the attention typically in our cachexia research, because we want to know at the business end what is going on. And certainly we've shown, and as I mentioned before, that there's plenty of factors that can cause wasting of these cells in these tissues. Well, I'm by no means the first to try to figure out what's going on with pancreatic cancer cachexia, and you may be familiar with this paper out of Mass General and the Broad, which was published in Nature a few years ago, where they looked at the KPC mouse. This is a KRAS-driven autochthonous pancreatic cancer model. And these mice develop at a very young age, multiple tumors within their pancreas. They don't live very long because of rapid growth and metastasis of these cancers. And the mice themselves do have something of a cachectic phenotype. They have lower fat and muscle mass. And this paper ultimately, I'm cherry picking some of the data here that I found are interesting. They didn't find any increase in any measure of energy expenditure in these mice. In fact, if you look at the relative VO2 and VCO2, they actually found that the tumor-bearing animals had lower metabolic output. And the respiratory exchange ratio was not dramatically impacted either. And then when they added pancreatic enzymes, they could restore some of the adipose tissue mass. There was no difference in skeletal muscle mass in this particular study, but then they found that those animals that they treated with pancreatic enzymes actually died faster. And they thought that perhaps feeding the animals was actually bad, or giving them more nutrition may actually have been bad for them, which I think would be really hard, hard-pressed convincing a clinician that that's the case. But ultimately, they came to the conclusion that wasting was secondary to exocrine insufficiency alone. And although it's a provocative conclusion, it certainly doesn't match our lived experience. But you can see where they came up with that, because if you compare starvation against cachexia, in starvation, we see a conservation phenotype, right? We decrease our metabolic rate, increase in hunger and appetite, more foraging behavior, and this all serves to preserve our lean mass. And this is, of course, nutrition sensitive. If you add nutrition, you can fix this problem. Cachexia has generally the exact opposite response, in which you see an increase, or at least a paradoxical lack of decrease in metabolic rate, a decrease in hunger and appetite, especially relative to the level of nutritional deficiency, lethargy, and less interest in looking for food, and lean mass is then catabolized, and this is generally nutrition refractory. And so it makes sense how they came to that conclusion, but it just didn't quite sit right to me. And so the question I wanted to ask first is this wasting simply a nutritional issue? We use a model of pancreatic cancer cachexia. It's becoming more popular. This one was developed by Dan Marks and involves the orthotopic injection of pancreatic cancer cells derived from the KPC mouse into the tail of the pancreas. We titrate this dose to get about a 2-week time frame that is very reproducible in its ability to induce anorexia starting about a week after tumor implantation, which is associated then with progressive loss of both fat mass and lean mass. So we feel like we have a good model here, and this model was published by the Marks lab, although they do it a little bit differently in JCSM a couple of years ago. And so what we went to then look at is to say, well, if this is, can we prove to ourselves that it's not just a nutritional issue. Can we modify nutrition and then find and control it and then find a difference? And so to do that, we generated a pre-cachexia model. So I told you that at day 7, the animals still are eating the same amount as their sham operated control group. They don't show any deficit in fat mass or muscle mass, here shown by the gastrocnemius mass. And yet, if we restrict their feeding to about 50% of their baseline food intake for, after the surgery, after they've recovered from surgery, so for the last 4 days of this study, what we can do is, although we don't have any impact on the fat mass in these mice, we do induce a change in gastrocnemius mass. And we've repeated this now multiple times. And so this is pretty durable finding. And so this tells us that these mice do have an increased susceptibility to nutritional challenge. Now, is that just the malabsorption that we heard about before? Well, Catherine Pells, who's a very talented former technician, now medical student, ran some thankless studies on the feces from these animals and looked at fecal protease activity and then total fetal protein and lipid content. And we see no difference in protease activity at this time point, or even a few days later when we're at more of a mid-cachexia phenotype. And you would expect to see more protein and more lipid in the feces if they had malabsorption. We actually see paradoxically less, and we think that that's actually because the mice tend to eat a little bit of bedding, and we think that may be some sort of anxiety-like behavior, or perhaps it helps them with some sensation of nausea. It's unclear why they do it, but one thing's for sure: we've not seen any evidence of malabsorption across multiple studies. So then what is causing the metabolic susceptibility? Well, is it hypercatabolism? When we look at, and this is just one example from indirect calorimetry, the RER, but we don't see any difference in RER. We don't see any difference in VO2, VCO2, and heat generation in this model at these time points. And when we look at the BAT, we're not really seeing a whole lot of change in UCP1 expression. When we go to the white adipose tissue, if you look at the markers of browning, you actually don't see an increase in this model, but you see a decrease in those markers of browning. So we're not getting that sort of hypercatabolic brown fat energy inefficient phenotype at all. And we are starting to see the normal signs of ubiquitin ligase expression with MAF and MRF and FOXO up, particularly in the mice when they are fasted at this time point. So we think that this hypercatabolic phenotype doesn't quite fit these mice. So the question then came, well, what is going on? And to think about that, I wanted to go back kind of to basic physiology and ask the question, how do we adapt to fasting? On the initial hours, and for a human, this can last a day or more, when we don't have nutrition available, we break down stored glucose in the form of glycogen, and then that provides glucose to the body. And anybody who's been on an endurance exercise trip knows the feeling when you bonk and run out of your glycogen, because it feels like all of a sudden the fuel's been sucked right out of you. So this is a limited resource, but a very effective resource. We then mobilize our fats, and through the process of beta oxidation, we're able to generate ketones, which can fuel both the brain and the body, as well as glucose from the glycerol backbone of fatty acids. And this basically is what keeps us going until we run out of fat, and is thought to help preserve our lean mass, which is finally, when we run out of fat, we start breaking down lean mass. And in that case, one of the main things that's happening is we're actually converting some of our amino acids here into glucose, specifically alanine. But and we're also liberating amino acids for use in protein synthesis elsewhere. So, this is the step that we're trying to avoid, and this is the step I think we're seeing in cachexia. And this is controlled by endocrine signals, to be sure. So insulin, glucagon, and glucocorticoids. You want to see sort of this would be driven by an increase in the counter-regulatory hormones of glucagon, glucocorticoids, growth hormone, and a decrease in insulin. And this helps us provide energy for work and maintain our blood glucose, and helps us recycle that stored energy in the form of fat, ultimately to preserve lean mass. The stages of starvation I kind of alluded to, and this is in a rat study from Goodman's lab in 1980. And what we see here is this is on the y-axis, the level of blood ketones. And you can see that during the earliest phases of starvation, the rats run out of glycogen pretty quickly. They start mobilizing their fat and increasing their ketone levels. And during that first stage, there's actually rapid catabolism of fat and muscle. And if you've done mouse studies or rodent studies and fasted them for 24 hours, it's pretty remarkable how much fat and muscle they will lose over that first 24 hours. If you do it for 48 hours, which is pretty hard on the animals, it's pretty remarkable how little muscle they will lose in the subsequent day. But during this second stage, the lean mass is really preserved. And we think that the fats are allowing that, because when we run out of fat and the ketones drop down and we enter stage 3, that's when you really start seeing breakdown of all the lean tissues. And so this really supports George Cahill's idea that the liver is the intermediary that is turning fat into energy resources to preserve muscle. So we wanted to look at that role of the liver in the case of pancreatic cancer. And if we just take a broad look at the glucose-related or carbohydrate metabolism in this case, we can do fasting or food restriction as a metabolic stress in these animals. And you can see that we in general see a slight decrease in circulating blood glucose in the tumor-bearing animals. And that effectively, both the tumor-bearing animals and the non-tumor-bearing animals are relatively similar in their ability to mobilize liver glycogen. But I will note that the ad-lib fed animals appear to have less glycogen stored up at baseline, telling us that they're probably accessing glycogen in an earlier stage or not storing the glycogen. And I think that that's an important note. But we then ran an alanine tolerance test, and just to familiarize everyone with that, alanine is transformed in the liver into glucose via the gluconeogenic pathway. This is part of the Cahill cycle, the name for George Cahill. And in this case, the alanine tolerance test, we give basically a pharmacologic dose of alanine to fasted animals to test the gluconeogenic capacity of the liver. You can also do this with lactate. The problem with doing it with lactate is you tend to change the pH of the blood pretty substantially, or the animal, when you give that big of a dose of lactate. So we went with alanine instead. What you can see is the cancer-bearing animals are unable to mount the same level of glucose response. So this tells us that this deficit in blood glucose is likely due to decreased gluconeogenic ability in the liver. Now, on the flip side, let's look at what happens with fatty acids. When we look at their ketones, when fed, ketone levels are quite low, but when we fast these animals, we really reproducibly see that cancer-bearing animals are less able to mount increase their blood ketones. And very similar to the alanine challenge, we did an octanoate challenge. In this case, we hit them with a large dose of free fatty acid octanoate in a fasted animal and test their ketogenic capacity. And you can see here that the ketogenic capacity in these, not only do they start at a lower level, but the ketogenic capacity normalized to their baseline is substantially reduced in the tumor-bearing animals. And so this tells us that there is a deficit both in gluconeogenesis and ketogenesis in the livers of these animals. Well, so the way we think about that is now that pancreatic cancer is somehow reprogramming the liver and blocking the adaptive response to low nutrition. Could it be just a hormone difference? I won't sit on this slide very long because the short answer is no. We don't see any difference between our cancer-bearing and sham animals at any time point with any of the regulatory or counter-regulatory hormones. The only differences we see are those induced by fasting, and those would be expected and are not different between our groups. So it's not hormones. And in fact, we see this evidence of this liver reprogramming conserved both in patients. If we took and we compared cachectic versus non-cachectic patients who were undergoing pancreatectomy, so these are fasting glucoses pulled preoperatively, and we see that cachectic patients have statistically significantly lower fasting glucose levels, not hypoglycemic, but lower than their non-cachectic cohort. And again, fasting ketones, in this case, we see a difference between our control blood. These are blood samples taken from our endoscopy suite. So again, these are fasted individuals who are coming in for EUS, and we collect blood at that time. And these are the ones who did not have any diagnosis, or any malignant diagnosis. And yet, when we compare that to our cancer patients or patients with pancreatic cancer, we see that just having pancreatic cancer, whether cachectic or not, decreases the levels of fasting ketones. Similarly, if we look at the KPC mouse, the autochthonous model, so effectively the same cells but arising sort of under genetic control naturally in these mice, we see a decrease in fasting glucose and fasting ketones. So we think that this is conserved across pancreatic cancer. We take livers from our tumor-bearing and sham-operated animals and did bulk RNA-seq, and we found a really pretty remarkable number of genes that were alternatively regulated between the groups. And most notably, I would say, we saw this big decrease in expression of genes associated with mitochondrial fatty acid beta oxidation. And this is, of course, the pathway that leads to ketogenesis and is the step preceding ketogenesis. And when these colors got changed a bit, so I'm going to have to walk you through this. I generated this basically to take a look at all of the enzymes that are involved in the process of beta oxidation and ketogenesis. And the blues here relate to this heat map coloring system. These sort of purple ones are meant to be gray. I think they got changed when I changed my background format, so sorry about that. But the point being that all of the genes in this pathway are either unaffected by cancer or downregulated. Similarly, if we look at gluconeogenesis, we see a similar pattern, which is to say that the gluconeogenic enzymes are almost all downregulated. And so we are not seeing we're seeing, at least on a gene expression level, evidence of impairment in both of these processes that are critical for maintaining the adaptive response to low nutrition. Well, that's all fine and good, but that's only important if the liver itself can control muscle catabolism. This is an idea that's actually quite old. About 50 years ago, I think it was written about for the first time by George Cahill. The idea that burning fatty acids was how you protected against muscle catabolism. This has never been directly tested, although there were some really neat studies done in the 80s where they damaged the liver, for example, with hepatotoxins, and found that then ketogenesis was reduced and there was increased protein wasting. And that protein sparing seems to be linked very tightly to the availability of circulating lipid fuel. So once you run out of lipids, then you really go to break down muscle. But we haven't had much evidence that directly links these processes to muscle physiology. And so if ketogenesis and a decrease in it was really important, we would expect that ketones would have some impact on skeletal muscle metabolism. And so we wanted to ask that question. We went into the well-known C2C12 myotube model, and we treated them with pancreatic cancer conditioned media from the KPC cells. And it really reliably causes this reduction in myotube diameter, which we see here. But what was really remarkable to us, and I'm only showing you one of several studies, is that when we add ketones, when we supplement this with ketones, we basically have a complete restoration of our myotube diameter. And that's associated with improvements in the typical E3 ubiquitin ligase genes. So it's mapping that somehow the ketones are actually reversing some of this catabolism in the muscle, at least in vitro. The question of how the ketones reverse muscle catabolism is open. Are they just a fuel? Is it an antioxidant or redox biology driven? Is this related to their role as HDAC inhibitors? And these are questions we're actively asking right now. But it does offer a link for us between liver physiology and muscle physiology, and sort of buoys our interest in the liver as a potential mediator of pancreatic cancer cachexia. So what is the nature of the signal from the cancer to the liver that causes this reprogramming? Well, I think everyone here has probably heard enough about IL-6, but I'll be darned, it just keeps popping up. And as we know, IL-6 is a negative prognostic factor for survival across a host of cancers, including pancreatic cancer. It's associated with cachexia and wasting in pancreatic cancer and other cancers. Really lovely work by Teresa Zimmer's that came out this year showed a complex interplay that IL-6 orchestrating cachexia through this interplay between the tumor, the fat, and the muscle. And interestingly, the liver itself, hepatocytes express IL-6 receptor. And some recent work by Greg Beatty's lab at Penn showed that IL-6 actually reprograms the liver to promote metastasis. So we know the liver is well suited to receive the signal from IL-6. We know that the levels of IL-6 are elevated in our tumor model. And when we look at that RNA-seq data, we're showing here in red all of the STAT3 target genes, and you can see that they're almost universally upregulated in the cancer-bearing group. So we think that IL-6 is certainly having a major signaling role on the liver. And the question is that important? Well, what we did to test that question was we used an IL-6 knockout. And I want to say first that our particular clone of these KPC cells does not produce IL-6, doesn't secrete it into the media, and they don't produce it themselves. So we know that the cells are not making IL-6. Now, when we put these tumor cells in an IL-6 knockout mouse, what we see is a reversal of much of the cachexia phenotype. We see that gastrocnemius mass is normalized. This does not normalize food intake, however. There's still a separation there between our sham and tumor-bearing groups. It did not normalize fat mass. And there was really no effect on corticosterone. I know that that's a common thought process. I believe Doug Fearon's lab did some interesting work in a pancreatic cancer model linking IL-6 to corticosterone, and we're not seeing that in our model. We don't really see much of a rise in corticosterone unless you fast the animals or you wait till very late in the course of cachexia. Not only that, but the fasting metabolite changes are largely reversed in the IL-6 knockout. You see the blood glucose is normalized. Blood ketones appear to be normalized between the groups. And when we look at some of the key genes that were downregulated in our wild type animals, they are normalized by the IL-6 knockout. And these are all gene expression within the liver as assessed by qPCR. So it looks like the IL-6 knockout animal is pretty refractory. And that would suggest that IL-6 is coming from a non-cancer cell source and is fundamental for this physiology. Well, where is the IL-6 acting? Is it acting in the liver? To ask this question, what we did is we generated some liver-specific STAT3 knockouts by crossing the STAT3 flox mouse with the albumin-Cree mouse. We used a 10-day study. So normally at 10 days, what we see is a loss in muscle mass in these mice. Whether you feed them ad lib or pair-fed, they're pretty reliably cachectic at that time point. We pair-fed to control for any differences in feeding. We found that these mice had the same rate of tumor growth. And yet when we looked at the gastrocnemius mass, as you can see here in the bottom left, we see that although the control animals lost mass, the liver-specific STAT3 knockouts had no separation. And similarly, even though these were not fasted animals, we actually saw a signal in our blood ketones, and that seemed to be reversed. Now, that really isn't the best model to look at ketones. So we did another study with these mice. And this, again, was an octanoate challenge. So we fasted the animals for 24 hours before challenging them with octanoate. And you can see that the fasting ketones, all these animals had cancer. The fasting ketones were improved in the liver-specific STAT3 knockout, and in response to the octanoate challenge, they were able to maintain their ketone production better than the control animals, suggesting that IL-6 is signaling directly on the liver and that this may actually have a direct role in driving cachexia. So the other part of this question then is lipid mobilization from adipose tissue. And I'm going to go through this pretty quickly so we can leave time for questions. But the main signal to the liver to turn on these processes is, in fact, free fatty acids that are liberated from the adipose stores. And we wanted to know, well, is that behaving normally? We've always kind of thought that that lipolysis was increased in cachectic animals. So is that indeed what's happening? So to ask that, the first thing we did is we just kind of looked at the fat mass in at a couple of different time points in our models. And you can see that if we control for food intake, which the graphs on the left more or less represent, because there's no difference in those groups, we don't see any difference in between cancer-bearing and sham-treated animals in terms of their white adipose mass. If we don't control for food intake, we do see a decrease in fat mass, and they seem to be equally susceptible to fasting. So there's not any obvious signal of enhanced lipolysis. And then if you look at the plasma non-esterified fatty acids, you can see here that there's an attenuated increase in fasting free fatty acids in these mice. So they still go up, but they don't go up quite as much as they did in the control animals. So this kind of flies right in the face of the idea of enhanced lipolysis. So we wanted to ask this question more directly. We used an ex vivo lipolysis assay in which you take the fat pad out of tumor-bearing or sham-operated mice, you put it in a culture dish and media, and then you can stimulate it with isoproterenol, a beta-adrenergic agonist that stimulates lipolysis. When we look at these fat pads and we do qPCR on them, you can see that the two primary genes that control lipolysis, adipocyte triglyceride lipase on the left and hormone-sensitive lipase or LipE on the right, both are substantially downregulated in both ad-lib and fasted conditions in the cancer-bearing mice. Furthermore, when we put these fat pads in the culture media, we see that lipolysis, which is measured by the accumulation of glycerol in the culture media, is suppressed in both the unstimulated and stimulated conditions. So this tells us not only do we not observe enhanced lipolysis, but we're seeing a defect in lipolysis in these mice. We then repeated a similar study using the NIH 3T3L1 cells, so an in vitro model where these were treated with KPC conditioned media or conversely just media that was conditioned in the 3T3 cells themselves. And when we compare the rate of glycerol generation in these assays, we again see a decrease in lipolysis in the conditioned media treated cells, and that is again associated with a decrease in the expression of ATGL and LipE. So this phenotype seems to be consistent both in vitro and in vivo. So in contrast to the prevailing theory that lipolysis is increased, and that's what's driving the catabolic state in the fat, we began to wonder if indeed it was actually a deficit in the anabolic potential in these animals. And so although lipolysis is down, the only way you're going to continue to lose fat is if you also can no longer generate fat. To ask this question, we used a fast refeed study. So we took tumor-bearing and sham animals, we let the tumors grow up to the point where we knew cachexia would be present. We then fasted them for 24 hours before exposing them to a 24-hour refeed. We terminated animals both after the fast and after the refeed, and with a primary endpoint of looking at adipose depot mass. The goal here was to see, so when we do this fast, we know from prior experience that we can get the levels, the weights of the fat pads to be the same between tumor-bearing and non-tumor-bearing animals. And then the goal here was to then see if the refeed itself would if these animals were able to add mass to their fat pad. And you can see both in the subcutaneous and visceral compartments that the fasting did exactly what we wanted. We see equivalent fat pad masses between tumor and non-tumor. But after the refeed, the tumor-bearing animals gained no additional fat, whereas the healthy animals were able to replenish their fat. And then associated with that, when we look at gene expression by qPCR, you can see downregulation of many of the main genes that are involved in adipogenesis and lipogenesis, including the key regulators CEBP alpha and PPAR gamma. We take this back to the in vitro model, the 3T3L1 cells, again treated with KPC conditioned media or control conditioned media. And we measure the rate of lipid droplet accumulation using an oil red O stain, which you can then quantify on a plate reader, which is kind of a fun way to do it. It passes the eye test. As you can see here, the KPC conditioned media have substantially fewer lipid droplets. And then when we quantify that, we see a significant reduction in their ability to generate lipid droplets. So we're fairly convinced that at least in this model, the fat wasting phenotype is associated more closely with a deficit in anabolic potential rather than an enhancement in lipolysis. When we take fat pads out of these animals and do bulk RNA-seq, we see about 600 differentially expressed genes. About 500 of those are upregulated and about 100 are downregulated. If you take this batch of downregulated genes and look at it, the hallmark that is most closely associated with the downregulated genes is that of adipogenesis. And indeed, the transcription factors that are most associated with that set of genes are the master regulators of adipogenesis and lipogenesis, PPAR gamma and CEBP alpha. On the flip side, when you look at the upregulated genes, the 500 or so genes, the process most closely related to that is an inflammatory response. And indeed, if you look down this list, they're almost all inflammatory responses. And when we look over here at the upregulated transcription factors associated with these with the upregulated genes, you see REL-A and NF-kappa B, both of which are key transcription factors as part of the NF-kappa pathway. So this really suggests to us that this is an inflammatory mechanism that is driving down adipogenesis and probably driving down lipolysis as well. And so our model at this point in time that we're working on still sussing out is that the tumor itself is releasing IL-6, which is inducing STAT3 phosphorylation in the liver that is then preventing both gluconeogenesis and ketogenesis and allowing for skeletal muscle catabolism, in part by the absence of ketones, which they themselves seem to be protective. We're currently working on some of the mechanistic steps to understand these links. And I guess the other point that I want to make is that the availability of fatty acids may be actually down from the fat via another inflammatory process itself. So in summary, pancreatic cancer cachexia is defined by this increased susceptibility to nutritional challenge. There's a deficit in adipose mobilization and oxidation. Adipose wasting is secondary to impaired anabolism and not increased catabolism. And IL-6 may actually be coordinating this hepatic reprogramming. To me, what this tells us is that there are definitely new ideas and new targets for reversing cachexia, and that we should be thinking both about improving anabolism in some patients, as well as perhaps targeting the liver as a major site of action. I'd like to acknowledge the work of all of my lab and all of my outstanding collaborators at OHSU and other places. I actually didn't have enough room for them in this slide. And our funding from the NCI and AACR, as well as the Knight Cancer Institute. And I'd really like to thank all of you for sitting here and your attention today, and look forward to any questions that you have.
Well, thanks, Dr. Grossberg, for a really wonderful talk. I really have not enjoyed having a pandemic, but I have to say, having the opportunity to have people like you in my office on a random Wednesday morning has definitely, in talks like this, has really been a positive for me, and I think a positive for our community. So thanks for a really wonderful talk. You are welcome to put your questions in the chat if you would like, and I will read them. We have a couple to start with right now. In reference to your liver RNA-seq data, Grant Hatch would like to know if you saw any changes in anaplerosis. And he's specifically wondering about what you think might happen if you had a hepatonic acid challenge.
Yeah, that is a really good question. So we did not track, I'm going to say that we kind of dipped our toe in that pond, but because other things sort of rose to the top, we followed those. And one of my collaborators, Rachel Perry at Yale, I discussed this with her, and she said she's really interested in that, but wasn't sure that RNA-seq would be very valuable interpreting that. So I kind of took my toe out of the pond, but admittedly, I'm deferring a little bit to her. So my disappointing answer to you is we kind of looked at some genes. There were some patterns, but not necessarily a consistent pattern of gene expression there. And I did not dig deeper, so I don't have a satisfactory answer. But the heptanoic acid idea is a really interesting one. And so I think you're going to force me to go back into the data this afternoon and take a look.
Funny how that happens when people ask you questions.
That's why this is so great.
Georgina Mancellini would like to know if you saw similar changes in other muscles, perhaps other limb muscles like quadriceps and tibialis anterior, as opposed to just if there's something special about using gastroc.
Nothing particularly special about gastroc. We always collect tibialis anterior. We don't collect the quadriceps muscle. Maybe we should. We haven't been. Part of that is because we tend, for reasons I didn't discuss in today's talk, we actually time lock our terminations. And so we really have to get through them in a short period of time, because we've actually found time of day really dramatically impacts some of the things we're looking at. So the quadriceps are on the sorry, didn't get it list. I suppose we could do that afterwards out of the carcass pile. So it's a good idea. The tibialis shows the similar biology. We tend to save those now though for histology, which I didn't show.
Yeah, I think we're all learning that we need to be a little bit more rigorous and careful and thoughtful about the time of day we euthanize our mice, and that there's substantially more variation than perhaps we have recently given credit to. I know in our lab, we're having to be a lot more careful and thoughtful about it. Mit Joshi would like to know, what about phenotype changes of adipose tissue into beige adipose tissue? You talked about you didn't really see evidence of browning. Do you have any evidence of more of a beiging or a shifting, any towards the browning direction?
So we did a limited analysis of that because the data started pushing us in a different direction. I didn't show all the genes that we looked at there. Basically, from a gene expression perspective, we do not see anything. In fact, we see quite the opposite. But now I want to put a caveat on that. If you wait until late enough in these mice, like if I get them real sick and they're at that sort of end throes of cachexia, we do see some changes at that point in time. That would support beiging, but not at the time points that I'm discussing here, the earlier time points.
Perhaps a later event and not necessarily an early and maybe driving event.
Yeah, and I want to be really clear about something else, because there's a few things I said that like maybe fly in the face of what some other people have done, and I don't want to, this is just one model. And when Teresa and I were talking about this, she has putatively the same model and it does not behave the same way. And so we're going to exchange cells and try to see what's happening there. I don't know how much of this is model specific.
Yeah, I was going to ask you personally if you think that your model has much metastasis to the liver, and if you think that if that is mattering in the changes you're seeing in the liver.
At the time points we're looking at, no, but eventually, yes. And so there's ways we can sort of, if we do a lower inoculation of tumor and let it go longer, we'll get more Mets. Or if we wait till later with this larger inoculation of tumor, we'll get Mets. But looking at the time point we're looking at, we see very little evidence of that.
Rosanna Piccirillo would like to know if you have any evidence of changes in leukemia inhibitory factor or LIF, and perhaps if a discussion about its relationship with IL-6.
Yeah, that's a good question. My first ever paper was on LIF, so it's like near and dear to my heart, and I love that question. Admittedly, it's on the list right now. We're looking at the best way to ask several questions at the same time, and we've kind of been saving up tissue. So my disappointing answer for your question is I don't know in this model, but I'm very interested in looking. And it's totally possible, right? Like there's been some really great recent papers about LIF and pancreatic cancer, and I guess I have not looked enough to see if LIF receptor is expressed in the liver. I don't really know the characteristics of that well enough, but it's on the list.
Yeah, I know as a fellow new PI, my ability to come up with questions is far exceeds my ability to address them the way I would like to. Dr. Zimmers has a question for you. She's going to unmute and ask you.
Hi, thanks so much, Erin, for giving this great talk. So I have maybe a perspective question for you, and then maybe a more specific question about the adipogenesis versus lipolysis. So the first question is, there's so many examples now where we see cachexia phenotypes in tissues other than skeletal muscle, or even in skeletal muscle, that precede wasting. And so we need to start thinking about the muscle wasting in cancer cachexia as really the final manifestation of that. And, and do as you've done and look way upstream. So that's kind of a perspective question. And then the second part was, of course, I was going to ask about the differences in lipolysis versus adipogenesis. And so how do you square a lack of adipogenesis with a patient who shows up with a history of 100 pounds of weight loss in 3 months?
Oh, yeah. So I'll try to answer those questions in order. From the perspective question, that's sort of the altar that I'm praying at right now that you bring up. I am certainly interested in the idea of muscle wasting being a distal event. And I think some of that actually comes from some of the data that your group has published. When I've looked at some of the RNA-seq that you guys published, and in just looking over it, it doesn't quite look like what we see in our rapidly wasting mice or our sort of IL-1 treated mice. And it keeps making me wonder, like, how much of this is sort of the quote natural muscle wasting that occurs when you're metabolically bankrupt, as opposed to the sort of perineoplastic or inflammatory muscle wasting that we spend a lot of time looking at. That being said, our models keep pointing towards direct impact on the muscle. So I don't know if it's a model shortcoming or a vision shortcoming or a little of both, but I'm very interested in that idea. Lipolysis adipogenesis, I'm not going to pretend for a minute that this is all cancer patients. And one of the big things is certainly that along with those huge weight loss, we typically hear about people's appetite being pretty substantially suppressed. That story is usually pretty hard to get out of them. You almost have to come backwards, start with weight loss and get back. And then finally them and their family members will note that, in fact, they have not been eating much at all. But I still don't think that accounts for it. And I suspect there is tons of active lipolysis. And I think the message I want to send with this is not that lipolysis or enhanced adipose catabolism doesn't happen, but rather that an impairment in anabolism does happen.
Absolutely. Yeah.
And should be considered also.
Thanks so much. Great answers. Thanks.
Do you have any evidence in your cancer patients that there's increased circulating lipid in them? I would presume somebody has this data, but to be honest, I can't think of it.
Yeah, there are a lot of data out there. We are actually right now putting together our cohort to do that, and we're just expanding it before we run the assays, because the data I showed you were really preliminary data, the human data, and we've got tons of samples, but we've optimized some of our sort of cachexia classification among our patients, and now that we've done that, we're kind of compiling the plasma. So I don't have a direct answer to that in terms of our patients, but a lot of people published elevated circulating lipids. And that would fly in the face of what I've seen in my mice. So again, is this model? Is it timing? Is it the million other things that we have a hard time controlling for in patients? I don't know.
Yeah, and I always wonder about, the anabolic rate of our rodent models, right? And the effect of starvation or, cancer-induced pseudo-starvation being bigger on them than it might be on a human, just from an evolutionary perspective.
Yeah, they don't really have much reserve at all.
Correct.
They go basically, which is kind of funny because you think about a rodent, the amount of uncertainty in their next food source is tremendous in the wild. So they don't seem to be well suited. I guess that's why they breed so effectively.
I wonder if it's just because tissue is so metabolically expensive to maintain, right? So they have more plasticity to cope with the uncertainty of the food source.
Yeah. Oh, that's interesting. I think about that a little more.
It's a pet hypothesis that I have no data for. So
I like it though. That's a good one.
Sorry, I lost my place in my. Fabio Penna would like to know about the age of your animals and if they might still be growing, and if that might perhaps be playing a role in your time-dependent effects.
That's a great question. It's actually the second time I've been asked that question today. I gave another talk this morning, an internal talk, and so our mice are sort of early adult mice. And there's a lot of data, at least in rats. I don't know the data in mice as well. It shows that actually older animals have better, have sort of better metabolic reserve in the face of nutritional challenge than the younger animals. The biggest reason we haven't asked that question is just the expense and the logistical challenge of maintaining animals until they get old. My lab just expanded, so we can start asking more questions like that. The pandemic caught us a little flat-footed and short-staffed, and we were not allowed to hire during those 2 years. So.
Yep, sounds very familiar. Fabio also says that he agrees that impaired anabolism is not something that we're spending enough time thinking about. Christina Wu would like to know if there are hepatic protective treatments available early in the cancer diagnosis, and also if there is a relationship between alcohol consumption and the chance of PDAC. And so if there might be some liver-related things there.
Yeah, I don't know about hepatic protective treatments. If you believe Greg Beatty's data that showed that IL-6 signaling to the liver was essential for liver metastasis, then you could make an argument that some sort of anti-IL-6 treatment potentially could be protective, at least against metastasis. And my data, and I guess Teresa's data, might suggest that it might even help with cachexia. So maybe that's something we should all put our heads together about. Alcohol is connected to pancreatic cancer. It's not a super strong connection, but there really aren't very many aside from a handful of genes. Whether that's from pancreatitis, though, or some other factor is a little bit less clear, because alcohol is heavy alcohol use is a risk factor for chronic pancreatitis, which is a risk factor for PDAC. And we think that's kind of the order of operations there.
Yeah, I know it's really difficult to tease out those interactions because they are a whole group of behaviors that tend to go together and are associated with PDAC. Georgina Mencellini would like to know what time of day you euthanize your mice and if there's a specific reason for why that time of day.
Do it in the morning. If you do it at night, you're liable to not see differences in the muscle. And so I didn't show these data, but here's your spoiler. We actually did round-the-clock circadian studies in the muscle of these animals, as well as the other organs. And all of our favorite targets that we look at in the muscle were normal. Well, at least the gene expression level were all normal during the nighttime when the animal is eating. But when they are not eating during the daytime, that's when you get this separation.
Interesting. I think that probably fits very well with some of Jim Carson's work too in the APC mid mouse of time of day and fasted and fed really have some differences.
Really
Lorena Garcia Castillo would like to know how long you treated your C2C12 myotubes with conditioned media to see atrophy.
I think we did both 24 and 48 hours. And I don't remember which one we settled on for the data I showed you, and I should know that. But I know we saw it at both. I just can't remember if the 48-hour was more compelling, so we opted for that, or if the 24-hour was easier, so we opted for that. I know we made one of those decisions. I can look that up, but it was either 24 or 48 hours.
And then we have one last question. Mit Joshi is curious about if you've looked at specific levels of plasma, things like cholesterol, LDL, free fatty acids, and glycerol. You know, if you've kind of done a panel, but it kind of sounds like you're headed that direction in your PDAC patients.
Yeah, so in the PDAC patients, not yet. Again, we're trying to figure out, we've been trying to figure out the best way to ask all the questions at the same time, with that resource being limited, or which questions are most important. In the mice, we looked at free fatty acids, and I think I showed those data. We did look at glycerol and triglyceride levels. Triglycerides are no different between, glycerol levels were really no different between tumor bearing and non-tumor bearing. Cholesterol I have not looked at, and it's kind of interesting because some of the RNA-seq data might suggest there would be some differences in cholesterol biosynthesis. And I've not looked at LDL.
And then I don't see any additional questions in the chat, so we'll try and be respectful of everybody's time, unless there are any last pressing questions. I don't see any hands raised. So thank you so much, Dr. Grossberg, for a really wonderful, stimulating talk and discussion, and for joining us. And we're thrilled to have you as part of the society, and as we try and grow. So thanks so much.
Well, thank you for having me. And thanks for all the great questions. That was a lot of fun.
Yeah, thanks everybody. We really appreciate you joining us. You should have a link soon for the Cancer Cachexia Society business meeting in October, and we'll see you again for more science in November.